Now we're going to jump into practicing some of the questions from the first section of the biology section of the MCAT. Let's go. Question 1. A biochemist isolates an amino acid with an isoelectric point, a pI of 9.74, at a physiological pH of 7.4. This amino acid is passed through an ion exchange column containing a negatively charged resin. Which of the following amino acids was most likely isolated? All right. So in this answer set, we have options including serine, lysine, leucine, and aspartic acid. Hopefully the one that sticks out here is lysine. Lysine is going to carry a net positive charge here, especially when it is dropped into a solution that is so much more acidic than its own isoelectric point. Lysine is a classic basic amino acid, and so since it will carry a positive charge, it will stick to this negative column. Answer choice B. All right, number 2. The formation of a peptide bond between two amino acids is a reaction that involves A dehydration reaction between the N-terminus of one and the C-terminus of another. B, an isomerization reaction at the alpha carbon. C, the formation of an amide linkage with the release of a water molecule. Or D, a hydrolysis reaction that is energetically favorable. What do you think? In this case, this is describing a classic amino acid polymerization, which is performed by dehydrating and removing a water molecule, forming an amide bond. This is best described by answer choice C. All right, question 3. A genetic mutation leads to the substitution of a proline residue with a glycine residue in a region of a protein known for its alpha helical structure. Which level of protein structure would be most directly affected by this change? A, tertiary, B, secondary, C, quaternary, or D, primary. In this problem, we are asked about something that specifically describes a disruption to what is either alpha helical or beta pleated patterns. This is the structure level referred to as secondary structure. It's not primary because it's not changing the order of the amino acids, and it's not tertiary because it's not discussing the later implications for how it will fold. Instead, it's disrupting the alpha helix. This is a direct indication that we're looking at secondary structure. Number 4. Which of the following interactions is the primary driving force for the folding of a globular protein in an aqueous environment? A, electrostatic interactions between charged side chains on the protein's exterior. B, the formation of covalent disulfide bonds between cysteine residues. C, hydrogen bonds between backbone amide and carbonyl groups. Or D, the placement of hydrophobic residues in the protein's interior. All right, so this is describing the reason behind why globular proteins form the kind of chaotic globules that they form when exposed to water. This is primarily driven by the hydrophobic residues that are present that enter the inside of the globular protein when it enters this conformation. This is best described by answer choice D, the hydrophobic effect. Number 5. A scientist wishes to separate a mixture of 4 proteins using isoelectric focusing. The proteins have the following isoelectric points, or pI. Protein A, 4.5, protein B, 6.0, protein C, 7.5, and protein D, 9.0. After applying an electric field to the gel, which protein will have migrated the closest to the anode? A, B, C, or D? All right, so this is describing a scenario where we want to know who is getting closest to the anode or the positive end. So we are looking for the isoelectric point for the protein that is going to be the most negative. In this case, the one that is the most aggressively acidic will maintain the most negative charge and therefore would be protein A with the pI of 4.5. it will migrate until it reaches something equivalent to a pH of 4.5 in that gradient. Number 6, the movement of dynein along a microtubule is an example of non-enzymatic protein function. This process primarily relies on which of the following? A, facilitated diffusion of solutes across the cell membrane. B, conformational changes powered by ATP hydrolysis to facilitate transport. C, the protein acting as a passive structural support element. Or D, binding to antigens to signal an immune response. All right. Dynein is a classical example of an ATP-powered motor protein. It is burning through ATP to undergo the conformational changes that it needs to facilitate this kind of transport. Number 7, the induced fit model of enzyme substrate interaction proposes that A, the active site of the enzyme is rigid and perfectly complementary to the substrate before binding. B, the enzyme lowers the activation energy by destabilizing the substrate before it enters the active site. C, the active site undergoes a conformational change upon substrate binding to achieve optimal fit. Or D, the binding of the substrate is non-specific and occurs at multiple sites on the enzyme. And of course, this is a perfect example of a description of a model which still isn't perfectly representative of the way a substrate and an enzyme interact. But the induced fit model is a little bit closer than the old lock and key model because it describes how the active site undergoes a conformational change upon substrate binding in order to achieve optimal fit. Answer choice C. Number 8. A drug is developed that binds to an enzyme at a site distinct from the active site. This binding event alters the enzyme's conformation, reducing its catalytic efficiency regardless of the substrate concentration. This drug is best classified as A, an allosteric activator, B, a competitive inhibitor, C, a zymogen, or D, a non-competitive inhibitor. And of course, the description that they're giving involves the inhibitor changing the enzyme's conformation and reducing its efficiency. So it is an inhibitor, but it's not binding at the active site, making it a non-competitive inhibitor. Answer choice D. All right. Let me know how you did, and I will see you in the next section as we go through the next topic in our complete guide to what is on the MCAT.